So, since Emily showed the beginning of the ride at the opening of the conference, I thought I'd show you where it winds up and how this 160 riders are committed. I got to know about the ride through the special relationship we have with this young man, who was the first, I think, person in the UK to receive gene therapy for Duchenne, Connor Crawford. And so his family has become a family to all of us at the University of Florida, where gene therapy was actually originally developed now 30 years ago. And it's been, I think, too slow a progress for my patients. We really want to accelerate the work in this area. And now it's a tough day, actually, in the US because last night Peter Marks resigned from the FDA and was one of our strongest advocates in this field. So hopefully we'll find a way. Maybe I'll just ask for asylum in the UK. Is that possible? So just to go through some of the basics, because many of you are new to this, the goal here, and since, as Volker so elegantly pointed out, in the beginning, we didn't even understand the genetic cause of Duchenne. We knew it ran in families. We knew there was a unique characteristics of all the boys that were affected, and it was unique and that it was only in boys as it was originally understood, and now we know that even females who have an imbalance of inactivation of one X chromosome can even manifest with Duchenne. But understanding the genetic cause helped us think about ways to replace or supplement that gene or even repair it. And so I'll touch on some of those things. And so at our institution, one of the first things that was tried is in the eye, where a genetic cause of blindness was being studied by one of our faculty called Leber's congenital amaurosis. And that actually program led to the first approval of a gene therapy. And that's what really accelerated the field, particularly the entry of pharma and biotech into this space, because as academic investigators, at least previously relying on the NIH mostly for our ideas and funding, needed industry to help bring these products to the market. But the idea is that a gene could be put into many cells and ideally all throughout the body. The retina was easy because it is a very localized space and required a very small amount of the gene therapy product. So over this past 20 years that we've been trying to improve on this, one of the big accomplishments has been how to even manufacture the products that can affect body-wide. And just to reiterate, the goal is to deal with the DNA that's in each cell that makes proteins. There is now also the possibility to directly edit the DNA within a cell. And in the case of Duchenne, that may mean removing a part where the errors are in order to put the gene back in frame, as it's known, to liken Becker muscular dystrophy, where a larger dystrophin is made than the ones that are added through this gene replacement strategy. And ultimately, there are means now to even manipulate RNA with editing or RNA delivered through lipid nanoparticles. So the two classes of gene therapy vectors that are used are either viral vectors or non-viral delivery techniques that have been more challenging to make work in the musculature. There's still a strong interest in this, whether it's through mRNA with lipid nanoparticles or other vehicles to get those compounds into cells. So far, the viral vectors have been the most efficient. And maybe to reiterate one aspect of what Volker said, the latest revolution in this part I'll touch on at the end is that through artificial intelligence and machine learning, modifications of the virus have been achieved to substantially increase their affinity for muscle cells and heart muscle cells. So I'll touch on that at the end. But you can see that the clinical trials in gene therapy have, in the oncology space, mostly focused on lentiviral vectors, which are used outside the body, and then those cells are returned. In some cases, to combat cancer, those cells that are engineered outside the body can go and eradicate cancer cells elsewhere in the body. And in fact, we're hopefully using this exact same technology to reset the immune system in cases where a patient may have received a gene therapy vector that was either less effective or at a lower dose in order for them to receive it again. So when people say, well, this is only a once in a lifetime treatment, in the case of a neuron, that's fine. It seems to live the life of the neuron. Those vectors that are entered into cells that help treat SMA seem to be lifelong, but in the musculature, this is not the case, and I'll come back to that. But the other vehicle that's used to deliver genes is AAV, which is where my focus has always been. AAV has been engineered to remove all the parts that are in the wild-type virus with the therapeutic parts. And we'll touch on how this actually works inside a cell. So the cells have receptors that are meant to bring proteins in and out of the cell, and this is where this AI modification now has had a big impact. Because once you can increase the amount of vector that binds to the cell surface, you can increase the success of getting those genes into cells or getting into more cells. And because muscle fibers have many nuclei, the goal is to get this gene therapy vector into a part of the cell that traffics through to the nucleus, where DNA is released from the vector. That DNA makes RNA, which is the intermediate that's used to make proteins, and those proteins are transported out to the cell and do their job. And in the case of the back one, there we go. In the case of Duchenne, the protein we're trying to make is really one of the largest proteins in the body. The limitation of the vectors that we had up to now is that they can only accommodate part of the Duchenne protein, and that's so-called microdystrophins. When we use this in vivo, that requires a very large amount of the viral vector to be given systemically. This hopefully will reach as many cells as possible, but we want to ensure also that that large exposure to the virus is safe. The inside part is made up of the gene components themselves and a way in which the instructions are provided to the cells to make that protein. Either can be directed at neurons, muscle cells, heart muscle cells. Those promoters are specific to the tissues that you want the protein to be expressed in. And for example, in Duchenne, one of the challenges is all the current products are engineered to work only in muscle cells and not, for example, in neurons, where we know there are some impact of the loss of dystrophin. That specific targeting is useful in some conditions, like it was in the eye, but we've wanted to also identify promoters that work in the important regions, particularly in Duchenne. So we're also focusing on how to make these vectors work not just in the musculature, but also in the brain. And of course, one of the challenges of using a virus given systemically is that our immune systems are designed to avoid those infections, particularly if we had any natural exposure to an AAV vector or ones that look like it. Our bodies would prevent this from being effective. And so these neutralizing antibodies that exist in the population, in fact, among adults, are about half of us have some level of neutralizing antibodies against AAV and would prohibit the use of AAV at least for the thresholds that have been set for some of the most commonly used vectors for SMA, for example, or even in Duchenne. And then when the exposure happens, there can be acute toxicities, and as was pointed out related to this recent event in a Levitas patient, those toxicities exist where the vector goes most, and that is in the liver. That has led to toxicities that can be counteracted somewhat, but it really cause us to have a very high suspicion for these problems, and we watch patients almost every day after gene therapy is administered. And then later on, many days or months past gene therapy, The T cell responses can be against the protein itself, and that's one of the things we've, I think, found a solution for that can eliminate the T cell loss of the vector in the muscle and hopefully improve its longevity. So the capsid part is the biggest burden that the body faces when exposed to the virus. And this is back to the idea that we can engineer more efficient viruses than have been used to date. That allows us to reduce the dose substantially. And in some cases, that's up to 100-fold less of the vector. So we studied a group of patients who had received AAV as part of a variety of different clinical trials, and then a second group that had been pre-treated with a drug that blocks the manufacture of antibodies in the body. And we showed that those patients that received only glucocorticoids had the most severe reactions and evidence of an early innate immune response that is due to proteins that prevent infection from parasites in particular, called complement. When we treated the second group of these 38 patients with a drug that blocks antibody formation, there were no significant antibodies developed, and the safety was enhanced. And now this approach is being used in a whole series of clinical trials, particularly in adults who are receiving gene therapy for cardiac disease. So what's the impact of this immune management and these new vectors for Duchenne? Of course, the timing of which these are delivered is going to critically impact the outcome. And we know that for all genetic diseases, particularly those with progressive nature, like in Duchenne, that earlier treatment would potentially lead to a better outcome. But also we have the possibility of growth and loss of the effect because of the increase in muscle mass. So we've got to pick a time. If it were given once, when is the optimal time before there's irreversible damage, or earlier on where we have to face the challenge of readministration? And I think this is now one of the more perplexing problems in Duchenne. We have this conundrum. Earlier treatment might lead to better, at least short-term outcomes, but is very unlikely to lead to greater durability. And now we have studies being done in children as young as 1 with Duchenne who receive gene therapy, and my worry is that that may have a short-term benefit, but not a long-term gain. So in fact, yesterday in Florida, where we have about 250,000 births a year, about half of that in the UK, so with 125,000 males, we would expect to identify 1 boy with Duchenne per week by newborn screening based on the genetics. And yesterday, a bill was approved in Florida to start newborn screening by whole genome sequencing. So currently, Duchenne CK testing, which is what's used in 4 other states, is not being done in Florida. But with this new whole genome sequencing, there would be about 600 different diseases that would be identified or verified through this approach. And I think the number one thing we'll find once this is underway is boys with Duchenne. Obviously, there are clinical findings early on, but at least in the US, the average age of diagnosis is 4.5. Those families which the mother is a carrier, that newborn, when identified, we will identify that about 1/3 of them will have a brother with Duchenne. So obviously early diagnosis is going to be really important in developing treatment paradigms, but it gets back to the immunology. So this approach, I think, will come. I think it's also being piloted in Europe, and we'll learn from both our experience and the experience here about how to use this information most beneficially. And the reason I bring up the issue of early treatment is that the vectors, as I showed you, they sit only in the cell in a part that is not part of the chromosome, so they're called extrachromosomal elements, and as new myofibers are formed, they don't carry the correction or the gene, and there is a slowly progressive loss of the effect. So that leads to where we are now. So that's the background of how gene therapy works and what has been accomplished so far with the products that are on the market. Volker pointed out the approval of ELEVIDYS in 2023, and then the expansion to all age groups. And it's an opportunity to really learn. The program began more than 8 years ago in the work that was done also by Solid and by Sarepta really has led the way in this, but now we've learned where are the limitations and where we can improve. So I'm going to discuss these 4 products that were actively in the clinic, and particularly what we learned from the Pfizer program. And just to remind you, this large gene has many functional parts. You all probably know where your son's mutation might lie and what is present in the vector relative to this. I'm going to show in this schematic. So these are the full-length dystrophin on the top and the large pieces that have been removed to make it fit in AV. So ELEVIDYS is the top one. You can see everywhere from the repeat 4 all the way through repeat 23 is missing. That, and then a portion of the C-terminal part is absent from almost all of them, except the Regenxbio product used a portion of the C-terminal domain, which they believe was beneficial in its function. So slight differences. So this is like shuffling around the parts. You can see that we have limited space, and each one of them has a large part missing. There's now, I'll highlight at the end, the potential to combine multiple vectors or use gene editing to make larger dystrophins that are more like the full-length one. So Sarepta's product has been studied, as was said, in a good number of people, the largest to date. Before this press release about the death, there were known to be 600 people that had received ELEVIDYS. I'm not sure how we got from 600 to 800 in a month, but a few really important studies going back to this 101 study that had 4 participants that are now beyond 6 years of treatment. A whole range of studies of different age groups with different endpoints, with an evolution of the vector to now a commercial process. And it's really the EMBARK study, the one in the upper right, that has been the foundation of the commercial approval. And at the MDA meeting a few weeks ago, 2-year data on EMBARK was published, and that's what I'm going to show you briefly to say where we are today with ELEVIDYS. So another unique thing about ELEVIDYS is based on a vector that was not being used by any of the other sponsors initially. This vector was discovered at Ohio State at Nationwide Children's by Phil Johnson. The differences may have important implications for safety, because among those that have received ELEVIDYS or an RH74 base vector, the adverse event rate has been lower than other products that have been tested. This product uses a promoter that directs the expression of the protein in muscle cells, but we have some concerns that this may not be as active in heart muscle cells as was anticipated in the beginning. So this is something still an open question, and this is very relevant, obviously, to the boys who are non-ambulant and would expect some benefit from a cardiac function standpoint. I showed you the transgene, and the goals were really to look at long-term outcomes in this EMBARK study, where half of the patients were treated in the first period and then the other half in part 2. That means that everyone in part 1 now has 2 years of follow-up, and Sarepta provided this kind of update, a very high-level summary of key functional endpoints. And so with the North Star, 2.8 points difference from change from baseline was felt to be significant. That's about at the level of which we would say it's clinically meaningful, however. That means that on only 2 of the tests you do to receive the North Star, someone might have preserved function and not be compensating. So it doesn't mean that it's been completely reversed, and as Volker pointed out, this is slowing the progression but not stopping it. And I think we have to maintain the goal of stopping this progression and not just accepting that we can slow the rate of progression. These other 2 secondary endpoints, time to rise and 10-meter walk-run, were favoring the treatment, but again, statistically significant, but maybe not clinically meaningful. And this was what was presented at the meeting, and I think it's an interesting set of conclusions. I added the ... because that was their summary statement at the meeting, that this is what we conclude so far. I'm not sure that I can accept the implications of the way this was presented, because even though we might get a slight increase in peak score at a given age, the notion is that we can completely change the natural history curve and maintain stability for years and years. And I think that's not what we're in fact seeing in patients. We're seeing this natural history curve, which we all know about from the great work done to establish this wide data set, is shifted to the right. It's not really changed its shape dramatically. But we should not give up the hope that we can get to what they expect would happen, that we would actually intervene at any stage in the disease. So the other program that's very far along is Regenix. As I mentioned, it's differentiated by having some additional bits of the dystrophin and the C-terminus. This base, again, as Volker pointed out, on a large deletion in a mildly affected Becker patient. The unique part of this, it has a relative of Rh74 called AAV8 that's driving the expression. Also, at the higher doses, appears to have a better safety profile than we saw with AAV9 vectors. This study is now open to boys 1 through 11, and criteria for entry for the younger ones are a stable dose of corticosteroids and greater than 10 kilos, and then older than 4, a specification for 100-meter walk without assistance and stable dose of corticosteroids. And so now the dose level has been selected as the middle box there, and that this will be studied in this larger group of up to 60 individuals. And the initial information that's provided is that we, at the second dose level, saw among 2 participants that were 8 to 11, between 20% and 75% of dystrophin expression. And again, this is where our goal should be, to get to complete restoration of dystrophin, not just in a portion of the cells. The third study that's active now uses a differentiated microdystrophin that has an important part of the function that helps autoregulate blood flow in muscle. This is called the nNOS binding domain. This is from repeat 16 and 17 on that previous graph. And we participated in this study when it first began with a different capsid that is being used now. So the original study, called Ignite DMD, used an AAV9 vector, but this exact same expression cassette. We've now studied those 7 boys more than 5 years, the 7 that were at our site, and find actually they have had substantial benefit from a cardiac perspective. Some of them have gained function from an ambulatory standpoint, but also as they've gotten older, we see the waning effect. And this is, again, really a goal overall in this field, is to reach the level of expression, or a plan from the beginning that there'll be a way to top off the expression at a later age in order to sustain the effect that was seen early on. Improvements in manufacturing of this product have helped improve its potency. And what's been reported at the MDA meeting was that of the first 3 patients of 6 treated thus far, there is evidence of substantial amount of dystrophin expression. Again, this same microdystrophin at the time points 8, 7, and 5 months after dosing. And this is the pace of enrollment has gone very well with this study compared to the original one, where it was stopping and starting with adverse events. And here is the outcomes that were shown in terms of the amount of dystrophin made by Western blot or another expression measure using mass spectrometry. You can see that there's near complete restoration of the amount of dystrophin expected if 100% of the cells were expressing dystrophin. It's a little hard to see in the lower panel, but this exact staining pattern that Volker described around the surface of the cell is hopefully enabling these cells to be protected from injury. And so the average number of fibers at this early time point was 78%. So about 25% more than was seen on average in the past. So this is encouraging, and this is in part due to the fact that this uses a novel version of AAV that's more effective in muscle. And I'll show the Pfizer study results, even though this program is not being continued, because I think we need to learn from it. So in this also very large study with 99 participants, 2-to-1 in the active group versus the placebo infusion, studied for 12 months initially, and then the delayed group was treated, so that in total the goal, and these boys will continue to be followed even though new ones will not be enrolled and the product will not pursue authorization. We will continue to learn from this cohort of volunteers that participated in the CIFRIO study and then the younger patient study called Daylight. So eligibility in this was slightly different, over 4 because functional outcomes were key, under 8 to try to achieve some homogeneity in the population, so they had certain characteristics in terms of their functional outcomes. And you had to fit between this sweet spot here of a North Star greater than 16 or less than 30. And these were the unfortunate, I think very disappointing to the community, results presented at the World Muscle Society. What we saw is that both the placebo group and the treatment group, during the acute period where steroids were increased to protect against the liver injury, they both improved. And I think what was unexpected is how that improvement would be sustained in the control group, such that by a year there was really no difference between those that received fortidistrogen and the control. This was, of course, Pfizer was very committed to this program. They were preparing for worldwide access to this product, because the initial results were much more favorable than this. So the shorter term outcomes were promising. And then once the study was unblinded and it was known that there wasn't a substantial difference at all, not only by North Star, but by even by age group, there was no difference whether you were under 6 or over 6, which was a key differentiating factor in the 2 groups in the ELEVIDYS study. So that led them to terminate the development of this program, and we will continue to learn why, even though there was a substantial amount of dystrophin being made by the vector. It's not as though the vector itself didn't do what it was supposed to do. It's a microdystrophin that did not have an effect in improving at least clinical outcomes. And so, they concluded that this just wasn't supportive enough, because you could say, well, why didn't they pursue the same course as Sarepta, which relied on the dystrophin expression levels for their approval? Pfizer wanted to differentiate this product from the beginning and insisted that the clinical outcomes be different and not just the dystrophin expression. So what's next? We've had the great pleasure to have support for this program from Duchenne UK for the past 3 years to understand how we deal with this problem. We studied now almost 120 boys over a 5-year period to look at their antibody levels pre-treatment. And these are a group of seropositive patients, and we see that really does not change. So it's not a transient issue, and certainly after exposure, it's even worse. So we propose to study various approaches to dropping the antibodies to improve eligibility of patients, or to treat those that were previously treated. So 2 kind of separate problems. And I'm just going to touch on 2 aspects of this. What existing drugs could we use that influence antibody production and or help deplete antibodies that are present? And these are the compounds we studied as part of our Duchenne UK supported program. I mentioned this rituximab approach as it really improves safety. It can also be used in conjunction with antibody lowering compounds to expedite the depletion of naturally occurring antibodies. It won't work once they've already been exposed, so this can't be given after the fact. And that's where I think we now, our next challenge is how do we address for those that have very high antibodies after exposure, how do we enable them to receive the improvements in products that are coming along. And then along the way, we began to study a new product called efgartigimod, which is made by argenx. It's used for treatment of myasthenia as an example of a drug repurposing. This drug helps block the recycling of antibodies, and therefore they're degraded faster. So I'll show you what we've done with this. So this is how the CD20 molecule works. It depletes both memory B cells and B cells that go on to become antibody-producing cells. You can see in the bottom row, those plasma cells are the ones that make antibodies. Once they've become plasma cells, though, they no longer have this marker and can't be killed by that antibody. So this is where these new technologies that use cells that were really products that were originally developed for cancer are now able to also eliminate plasma cells. And this is what we learned, that if you immunize a mouse with an empty capsid, and we start one of these immune suppression regimens, and here I've just laid them all on top of one another, you can see that with the combination of CD20, sirolimus, and another drug that blocks the B cell function, we can get almost 100% change in antibodies over 3 months. If you then add the Vyvgart to this regimen, you can deplete the antibodies even further because it accelerates the decay. So that's a program that we now are bringing to the clinic with support from some US foundations, where we would enroll 12 boys who were either have naturally high exposure to antibodies or ones that had previously received gene therapy. And we'll see what the effect is on antibodies by that method. We're also collaborating with 2 different groups to come up with very specific ways to degrade only AAV antibodies and not all antibodies of those other drugs. So this uses a trick that actually drags the antibodies into a degradation pathway in hepatocytes, and I'm very encouraged by this concept. The first data of a pan-IgG lowering drug that Biohaven developed has a 90% reduction in antibodies in just a few administrations of these drugs. So now we're making versions of this that work only on AAV antibodies. And then we're trying to optimize what goes into the vectors that could be used in conjunction with this. This was the original effort to reshuffle some of the parts in a microdystrophin. And you see, even just by reorganizing some of the domains, we can more than double the expression level. We take this one step further, actually, and not use just one vector, but 2 or possibly even 3 to replace all of dystrophin. This is the recent 2 publications from the group in Washington and in Indiana, that we can actually use AAV vectors to deliver full-length dystrophin. Or in the case of a 2-vector product, it will have 2/3 of the full-length dystrophin. And this is really going to be encouraging, I think, to restore all of the C-terminal domain, all of the parts that help regulate blood flow in the musculature. And we do it in a way that will actually reduce cost and use the capsids that I mentioned that will increase efficiency. We now have identified new capsids that are up to 20 times more effective than RH74 or AAV9, and those are required to bring these near full-length or full-length dystrophins into the program. And so I'll end by saying we're really learning a lot from commercial therapies. We're able to renovate this space where all of our commercial therapies are now given, and we get the support from the hospital to do that. It just opened last week, and so I think we have a team very dedicated to advancing gene therapies into the clinic for really to benefit Duchenne patients. And I'll just say at the end, I think we're going to save questions for the last part, but I will put out an ad. Anyone who sees your local pub that serves Tonebridge Brewery brews, there's a Connor's Ruby Ale will be served there starting next week, and all the benefits from the sale of the Ruby Ale will go to Duchenne UK. So a little advertisement for Tonebridge Brewery. Thank you.